today I will talk to you about a range of behavioral experiments I've been doing to investigate animal personalities or consistent behavioral differences and collective behavior of sticklebacks. So I've been fascinated by how animals live and move in groups, and social grouping is omnipresent in the animal kingdom, from flocking birds and schools of fish to herds of mammals and primate troops. And what is particularly interesting is you get these spectacular collective patterns emerging somehow from individuals within them. And so we really need to try and understand, how do these collective patterns emerge? How do individuals play a role in these groups? How do they make collective decisions? And how do groups function actually? And so there's been historically a strongly mechanistic approach to understand this collective behavior using a self-organizing approach. And so what research has been doing is combining experimental work and a lot of theoretical work, really trying to unravel these universal rules underlying collective behavior. And what has really become apparent is that we can actually understand a lot of collective emergent properties, from coordinating schools to collective decision making, from simple individual rules of simple agents responding to each other by aligning, attracting, and repulsing from each other. And just from these simple rules at the individual level, we can explain like these hugely seemingly collective patterns of animal groups. But one important point here is that a lot of this research has considered that groups are kind of homogeneous. They consist of agents or individuals that are the same, use the same interaction rules. But it's now really time to really add another layer of understanding, because as we all know, animals are not all the same, yet they still group together, and individuals might be of different ages, different sex, size, etc. There's this large phenotypic variation among grouping animals. So really the next step is to understand, how does this individual heterogeneity affect collective behavior? This has really been my research focus since my PhD. And so with this image, I will show kind of what is my research focus. So there's phenotypic variation among animals. This is hierarchical, strongly linked, so different phenotypic traits might be related to each other. This ultimately will affect individual behavior. The individual is part of a group, thereby it has an influence on the group, and the group has an influence on the individual. So there's social feedback there. And ultimately, this group is part of a larger community or population of animals with potential fission-fusion dynamics and among group level structure. And this needs to be considered in the environment, of course. So you have the broader ecological and evolutionary consequences of this individual level heterogeneity in animal groups. And my research is really trying to understand more generally among species, how does this work? And specifically, I've been using three-spine sticklebacks as a model system to understand this. So since my PhD, I've been doing a wide range of experiments, mainly in the lab, to really unravel the role of individual heterogeneity in collective behavior. And so because I want to understand what is the role of individuals in groups, I need to test a lot of individuals and groups. And so I need a high throughput approach to understand behavior at individual and group level. And for that, I developed a variety of systems that enable me to test a large number of fish repeatedly over time and across different contexts. Then to really help automate this, I've been using these Raspberry Pi computers, these low-cost single board computers, and written specific software that enables me to calibrate and set up all my cameras, and then have automated recording of these assays. And so I can test tens of fish at the same time and then run hundreds of sessions per day, so that in a couple of days I know multiple traits of all these different fish before I test them in different groups. And so then to further automate this, I wrote tracking software to really get accurate individual and group level behavioral characteristics. that really enable me to answer the questions about what specifically matters at the individual level for the group level behaviors. And so in my PhD, I was very interested in this phenomenon of animal personalities, which I nowadays really just like to talk about consistent individual behavioral differences. But the key thing here is that if we have animals that are different in terms of, or the same in terms of their size or age or sex, but still in a lot of species, we find that animals may be consistently different in their behavior. And so some individuals might be shy and others might be bold or sociable and unsociable. And we find this is repeatable over time in a context. And so what I was particularly interested is to understand what role do these behavioral traits play in group behavior. And so I used a classic assay to measure boldness. So fish were exposed to this rectangular environment. They had a safe area, deep area, and an increasingly shallow open area. And we automatically tracked them and measured how much time they spent out of cover, how fast they were out of cover, etc. And then we did this repeatedly over time. And we found that some fish were shy. They went little time out of cover, and others spent a lot of time out of cover. And it did so repeatedly over time. So there was high behavioral repeatability in this behavior. Then importantly, this trait was found to be strongly linked to foraging motivation. So when we actually fed the fish in their home tanks as much as they wanted, we found that even fish of the same size, that these bold individuals would sometimes eat almost twice as much food as a shy fish of the same size, even if there was no risk around. So this really highlights this boldness is strongly linked to some kind of foraging motivation. And then to measure sociability, we used the classic assay, which consists of this large open environment with two side compartments, with one containing conspecifics. And then by tracking the fish, we can see how much time does this fish spend near the others. And very sociable fish spend a lot of time near the others, and less sociable fish keep on exploring more of the small environment. Again, we find by repeatedly testing individuals, this is repeatable. And then this is also strongly linked to speed. So fish that are highly sociable, they stay near the others and don't move a lot, while less sociable fish tend to move a lot and have higher movement speeds. So then we test for this particular experiment I will go through now, we tested 125 fish twice on each assay, and we find that boldness and sociability were not correlated. So we had shy fish that were sociable and shy fish that were non-sociable and bold, etc. So we had a very nice mix of behavioral types that we then subsequently, I will go through this first experiment. I will go through the experiment later. But before that, I would like to just show you one more experiment about the plasticity of boldness. So by testing a lot of fish repeatedly over time, we got the impression that bolder fish were better, more predictable in their behavior and changed less in their behavior. So using our automated approach, we tested 80 fish every week across a 6-week period to see how does this boldness behavior change over time. And what we found is that these bolder fish that were initially exploring a lot, they didn't change much in their behavior over these 6 weeks. While these shy fish that were initially spending a lot of time out of cover, they increasingly spent more out of cover, even so that after 6 weeks, in general, they even spend more time out of cover. And so if we then look at the residual variation in behavior, we found that these shyer fish were much more unpredictable. Sometimes they spent a lot of time out of cover, sometimes they didn't. And so this really nicely showed that this boldness is linked to the flexibility or plasticity in behavior. Also during my PhD, I did a lot of experiments with pairs of fish. Pairs, of course, also a group of animals, and so helps us understand collective behavior in the most simple way. And we did this by having two boldness assays side by side with a transparent partition. So we could see which fish was leading, how they were attracted to each other, and how they influenced each other's behavior. And so I will very quickly go through these results. So the main thing that came forward is that also in a social sense, bolder fish were less plastic. So they spent the same amount of time out of cover when they were alone or with another, while shy fish increasingly went out of cover with another individual there. Again, bolder fish were much more consistent in their behavior, and shy fish were less predictable and had lower repeatability. Then looking at these transitions of going in and out of cover and who was leading these movements, this was really primarily the bolder fish, with shy fish very good at following. So it's the moment the bolder fish would go out of cover, the shy fish tended to follow. So there was a very clear leadership effect linked to boldness. And bolder fish were less socially responsive. So they would, when they would both be out of cover, the bold fish would be swimming in and out of cover, while the shy fish was really trying to be near the other, swimming near the partition. Then finally, we tested fish repeatedly with different partners. And then what we found is actually that these bolder fish were actually mainly driven by their previous experiences, their own behavioral type, while the shy fish were very plastic and their behavior could be well explained with who they were swimming at that moment. And so these range of paired experiments showed some really interesting insights about this boldness trait and grouping behavior. But then I really want to see what happens in freely schooling fish, where we can really disentangle collective behavior properly. And so here I will continue what I said previously, is we tested 125 fish on these two traits, and then we created 25 random groups of fish. So these groups had a wide range of compositions that we then tested repeatedly in a circular open tank. And this tank either had no foraging patches or refuge, or it had food patches, or it had also a safe refuge in the middle. And then we tracked their behavior over time, tested these groups repeatedly over a week, repeated in these contexts, and looked at the influence of these traits on collective behavior. And so what first came apparent is that sociability had a very strong influence on the collective behavior of the groups. More sociable fish were closer to others in these groups, and thereby they were more in the center of the groups, while these less sociable fish were more on the periphery and further away from others. Looking at the spatial positioning front to back, so in terms of the movements of the group, less sociable fish were much more likely to be leading the groups. They were more in front, with more sociable fish in the back. And if we then look at the propagation of turning changes, so one fish is making a turn and we can see how correlated are the movements of the other fish, this was also strongly linked to sociability, with more sociable fish being much more likely to copy what their group mates were doing. And by looking at these, how these changes of movement propagate through the group, we can create a leadership network where we can see that the flow of information, the flow of movement changes is generally going from less sociable to more sociable fish in the group. And then if we go and look at the group behavior, we first of all look at the key traits linked to schooling behavior, which is cohesion, alignment, and movement speed. We find that if we consider the group's composition on average, so we categorize them as less and more sociable groups based on the mean of the sociability types in the group. We find that these highly sociable groups, they're highly cohesive, they're strongly together, but they're not very well aligned and they don't move very far. They tend to be swarming, they are together, while these less sociable groups, they're maybe less far, they're further apart, but they are moving at a significant speed, and to stay together, therefore they're highly polarized. They have a high coordination in their behavior. Then by testing these groups repeatedly over time, we find that actually also there's high repeatability at the group level. So again, these groups were randomly composed. Fish could just swim with each other when they were in the environment. After each trial, they were put back in their home tanks individually. But still, when they saw the same group mates for these short periods of time in this environment, there was high repeatability. So some groups were always faster than others. Some groups always had clearer leadership, were more aligned. And this was very strongly linked to these individual traits. So this shows that the group level behavior really emerges from the individual behavioral types. Then finally, for this experiment, we also want to see what are the functional consequences? How do these traits affect individual and group behavior in terms of food intake? Well, here what came apparent is that it's really the combination of boldness and sociability that drives the foraging performance. So at the individual level, it were those fish that were relatively bolder than their group mates and relatively less sociable that had the highest food intake. And if you remember what I talked about previously, boldness is strongly linked to foraging motivation. So this explains that why boldness is positively linked with food intake. But then this sociability was negatively linked with speed and strongly, of course, linked to the social responsiveness. So these fish that are less sociable, they tend to lead more and therefore also find food more. Then if we look at the group level, also these two traits again explain how quickly these groups find the different food patches and depleted them, with the boldest groups with the least sociable individuals finding and depleting the foraging patches the quickest. And then ultimately, we combined these experiments with agent-based modeling, where we generated heterogeneity in terms of speed and goal-orientedness to reflect these differences underlying boldness and sociability. And there we could qualitatively provide a parsimonious explanation for these findings, that indeed speed and the tendency for finding resources can really explain this spatial positioning, leadership, group cohesion, and group functioning of the stickleback groups. And so last year I've been further doing experiments on the topic of these individual level behavioral traits and grouping behavior, but then at the next level, so among group structure and dynamics. And so I've been testing larger groups of fish in larger environments, developed special tags that we can, small backpacks we can put on the fish to recognize the fish over time, even when they go in and out of cover and across long periods of times. And I'm currently still analyzing this data set. And another experiment I've been doing is doing predator-prey experiments to see if specific behavioral types are more likely to be predated in these groups, and if that depends on the group composition. So bolder fish that are less sociable might have higher food intake in these groups, but are these fish not also most likely to be predated? And what if you are a bold fish in a shy group versus a bold fish in a bold group? So really understand more this social selection component that is related to these traits. Then finally, I would like to go through one experiment where we looked at the role of parasite infection in collective behavior. And so as many of you, or I think everybody of you will know, the sticklebacks are prone to a huge range of parasites. And the schistosyphilis is this fascinating parasite system where the stickleback is an immediate host. And we find that in our population in Germany, sticklebacks could have a parasite mass of at least 50% of the body. And so we wanted to see how does this affect individual behavior and group behavior. And so we looked at 120 fish, and we couldn't kill them before the experiments, but we predicted the parasite loads based on accurate photos with which we were 95%, more than 95% correct. With fish having 1 to 3 parasites, and indeed up to a parasite load of 50%. And so then we exposed these sticklebacks individually to an open environment, and we tracked them accurately again and looked at their movement patterns. And what we found is that these parasitized fish were generally much slower moving. They had a lower cruising speed and a lower maximum movement speed. But interestingly, they were more likely to keep moving. They even when they slowed down, they never really stopped. While the non-infected fish, they really tended to move more fast and then stop, fast, stop. So they had a very different motion pattern. This was very clear in terms of the speed and acceleration. But also if we then look at the turning behavior, and so So the left plot here shows in red the highly infected fish and blue the non-infected fish. And highly infected fish took longer to turn in their behavior. And this was clear when we looked at autocorrelations of behavior. And what this basically means is that if you look at behavior at a certain time point and then see how well this fish's own behavior is correlated over time to that time point, we see that the behavior of infected fish stayed more similar over time, so was more predictable. So in other words, parasitized fish move slower, keep on moving more, and are more predictable in their movements. Then finally, when we actually chased them to see how fast can these fish swim, they were able to swim much faster than they did in this open environment, but still it was considerably lower than the non-infected fish. Then we created pairs. We tried to create equal pairs of both infected, both non-infected, and a mix of fish. We tracked their movements accurately over time, and then we looked at their social interactions. And what we find is that, first of all, very clear effect was that these infected pairs were much slower than the non-infected pairs, as is logical arising from the individual speeds. But then these infected fish were also much less cohesive. They tended to be almost twice as far apart. They were much less aligned. So they were not as able to stay oriented together. They were not very well coordinated. So when one fish would change its movements, change its speed, these changes were not very well copied by the other partner. And this was again strongest in the pairs where both fish were infected. And then finally, if we consider the mixed pairs, we find that of those 20 pairs, by far the majority was led by the non-infected fish. And then we combine this again with agent-based modeling to understand what is now the driving effect here. Because we know parasitized fish are slower, they keep on moving more, they turn slower, they show less stop-go behavior, and they show lower social attraction, which could be both intrinsically driven or because they are not able to do so. And so then we generated models with these different variables. And I don't have time to go into detail here now, but what came forward is that the really important variable here is the movement speed. And so by the fact that this parasite strongly affects the movement speed of the fish, it has all these clear effects on group level behavior. And so with that, I would like to thank you for your attention, and I'm happy to answer any questions.
Thank you very much. And you have question in your chat.
Okay.
I think that it will be easy just to directly respond to them.
Okay, so the first question is, as I understood, in each group of stickleback there is a leader, isn't it? Is it consistently the one, or can it change in time and another fish becomes a leader? Yes, so there is, the key thing here is that leadership emerges from the relative differences among the fish. And so if we look at their boldness, bolder fish tend to lead more than shy fish. But this depends on the context. If we expose the sticklebacks to an open environment where there's no refuges, there's no food, these fish would just swim together, and boldness doesn't influence leadership here. But sociability does. So less sociable fish tend to lead more. But of course, that doesn't mean they're always the leader. And it's not that they are an intrinsic leader, because if you then have this less sociable fish and you put it in another group where it's suddenly not so, where it's suddenly relatively more sociable, then it will follow more. So it's really an emergent property that is relevant to the individual fish. And then the second question was, do you have data on the effects of parasites on courage behavior? Individuals in the experiments were not divided into groups of healthy and infected, for example. Wait, was this question asked before or after the schistosyphilis experiment?
Yeah, it seems to be before.
Was before, right? I guess, yeah. Because then I showed, well, I mean, our experiments were indeed like in an open environment with the schistosyphilis. And the key thing here is that there's been tons of research on schistosyphilis on risk-taking behavior with parasitized fish showed to be taking more risks, approaching predators closer, etc. But there's been very little data on actually the movement patterns like we did here. Like here we use the tracking data, the orientation of the fish to really know where fish are going. So we know that infected fish are bolder, but we now also show they are slower and less mobile, and that it affects collective behavior. Okay, question
Dmitry,
from
okay,
June.
Dmitry, are you okay with the response?
Yes, okay.
Yeah.
Shall I go? Oh, Pino! My dog is barking, sorry. Okay, so June has a question. What ecotype did you use for studying the collective behavior? It would be interesting to see if there's any difference between ecotypes, such as between marine and freshwater ones. Just a second. Pino, comments? Yes, great question indeed. So these, I didn't indeed provide too much detail about the backgrounds of our experiments, so because of lack of time, but all these fish I've been studying both in Cambridge in the UK, where fresh water from streams we collected, and in Germany, where with fish from the river as well as the Bodensee, a huge lake, and these are very different ecotypes indeed. So in Cambridge, they are standard freshwater sticklebacks, but in the Bodensee, the sticklebacks are very similar to, I think, marine types. So they have a much stronger body armor. They are 10 centimeters in size. We had many individuals that were at least 3 years old. And so what we try to do in these experiments, I showed at least that we used fish from the river that were more similar. And we wanted to do a comparison study with the more like pelagic types, but we haven't done that yet. But indeed, this has been on my agenda to further look into, and I mean, especially also with your your expertise on genetics. I think there's a lot of work to do there. Like I've always wanted to collaborate with people that have this knowledge, because my expertise is behavior, and of course behavior is driven by genetics. So it is important to better understand the extent that these effects are genetic or plastic. So yeah, I think there's, do you have a response? Yeah, I see. But yeah, let's keep in touch. It would be nice to catch up on that as well. I see one more question, so I just quickly go through that. 2 questions by Ekaterina. Did you try to carry out similar experiments in open waters? So you mean if we did these experiments in the wild, right? Yeah, so I mean, I would love to, but it's of course always a trade-off, right? So in the lab, we can get high throughput data on individuals and groups. We can get very detailed data on the fish, and a lot of experiments I haven't shown here, like we can calculate the field of view. We're doing these predator-prey experiments at 120 frames per second, so we can really disentangle exactly what is happening. But yeah, so I've not done any published work yet on sticklebacks in the field where we study the individual behavior and group behavior. But again, like the main reason is a trade-off between time and how well you're able to answer your questions. Because yeah, I think we need a lot more data on the field situation, and it seems you have a great team there in Russia with a lot of field work. So that's why what I also said previously, like if you get more video data, and to follow up on that, I'm actually starting a new job here in Spain next week, where I will be setting up a range of studies, including on sticklebacks here. And it's very fascinating here because the sticklebacks, here are completely different from Northern Europe. They tend to only live 1 year, and they are exposed to these severe droughts. So the Mediterranean rivers here, they dry up a lot, sometimes completely. And so the sticklebacks have this very high turnover rate, and a lot of them die every year. And actually they're now, extremely endangered, and there's conservation effort to save them. While you are catching, I see like nets with hundreds of kilos of sticklebacks. So it's completely opposite. And I'm working with an NGO now that is trying to save the stickleback from extinction here in Spain. And so I think that will be very interesting the next years to see what kind of field work we can do there. And then, another question there. I want to figure out how you are planning to use small recording tag. Do they affect fish movement or not? Right. So we've been using small tags on the spine of the fish. I think also like some of you have been doing. And we did some side experiments that show that the tags have a small potential small effect on the fish, but this doesn't have any influence on any of the behavior work we've been doing. And all the fish have the same tag on their spine, but still, that's also why we've been trying to do more experiments without tags. And then we developed these 3D printed backpacks. And so it's not on the spine, so there's no water kind of flowing against it, but it's very, it's like a elastic band, and you put it over the fish, and it sits tightly, and it's on the body, and it doesn't influence the dynamics so much. And then we can remove the tags as well. And so some pilot experiments showed that had very little influence on the fish's behavior. So I think there's a final question. Was any aggression against the infected fish noticed? Zebrafish are sometimes aggressive to the aged or sick individuals. No, not at all. I mean, in general, I rarely observe aggression in the sticklebacks, and this is also I try to not test fish in environmental conditions that resemble breeding conditions. So higher temperatures and more light, you get the sex differences become apparent, and then their behavior will become very different. So we tested fish particularly in lower temperatures and 12-12 light dark conditions, and we didn't notice any aggression, and also no reason I would think why fish would be aggressive to infected fish. So if you observed that behavior, I would be interested to hear more about that. Yeah. I see one more question. I don't know how much time there is.
Still has some, yeah.
Good. So yeah, I cannot pronounce his name in Russia. It's the previous I don't know the letters, but if this person wants
in Arabic, you need to pronounce it. Okay, it doesn't matter.
No, but I mean, if this person wants to respond later about aggression in zebrafish, then I'd like to hear.
Okay.
Yeah, and then Ahmed said, did you take into consideration the changing behavior in the lab than in the wild, affected by your movements in the lab, for instance, size of pond, etc.? Wait, so I guess do you mean like, do I take into consideration that it's an artificial environment, that we test animals in a lab environment, which isn't the wild situation? Yeah, like any person interested in animal behavior that wants to try to answer questions while minimizing other influences, there's this trade-off. You can do stuff in the wild and in the lab, and both have their advantages and disadvantages. For us, the lab approach is really the only way to answer these questions, but we try to minimize all these other influences by acclimatizing the fish to the lab for a long period of time. They're socially housed. They get used to the feeding. They get used to the handling. We expose them to the experimental environments, etc. So we try to minimize any artificial influences in their behavior. And then in these essays, we also try to make it as natural as possible, while still being able to track the fish. And so if there's like sand on the bottom or refuges or so, and that is possible, then we will include that. And we combine this as well with mesocosm experiments, where fish are outside in concrete basins with sand and the elements, etc. But then already it's much harder to get video data, for example. So I hope that answers your question.
Yeah, maybe I have one question. So the next presentation will be a bit shorter because Johan Eckloff, he is not participating in person in this meeting, and he only sent us a pre-recorded file, and it takes 25 minutes, so maybe a couple of minutes more. So I have a question. As far as I understood, so you are saying that the school is a system, quite complicated system, based on personality of each fish, of each member. And my question is it possible to measure fitness of the school, not fitness of individual fish, but kind of fitness of the school in terms of vulnerability to predator or effectiveness of food consumption, something like that?
Yeah, so as
because in fact the school is a unit which is operating in nature, but not individuals.
Yeah. I mean, so in nature it's a combination, right? You have also, depending on the habitat, etc. But the schools that I showed in my experiments, from that experiment where from Cambridge, they live in streams. And my observations in the wild show fish are sometimes by themselves, sometimes in pairs, sometimes groups of 20 or so, rarely more than in the tens, maybe 50. But in these streams, they don't school in much larger schools. But they definitely are also often by themselves, for example. But then in terms of group performance, so as I showed in one of these experiments, we looked at this functional behavior in terms of how quickly the groups were able to find all the food. And so we had this meter big circular environment with a refuge in the middle. And then we had these food patches around with bloodworms that the fish cannot see because they're the same color as the background, but they only could see it from about 5 to 10 centimeters distance. And so these fish really had to explore and then find it. And then there's this trade-off, like, do you stay together and then find a patch together and then eat it and then find another? Or do you split and then everybody finds it by themselves? And what came apparent there is that there were large differences in group performance in terms of how quickly all the groups depleted the patches. And then the groups that did the worst were those that were composed of very shy fish and composed of fish that were highly sociable. So these groups are basically all hiding together in the refuge, or when they go out of cover, they're all together swarming, so they don't go anywhere. And the bolder, less sociable groups, they explore a lot, they split more, they find patches and thereby deplete everything. And then the other functional consequence is indeed predation risk. And this is an experiment like the slide I showed with some photos of ongoing work. So we did an experiment last autumn where I did something similar. So I had groups of fish that were or bold or shy or all these mixes of compositions. And then I exposed them to pike in the lab. And then I saw what groups were able to survive longest with a pike and which individuals in the groups were more likely to be predated. And so in this way, we can look at both social selection, so how does an individual's fitness depend on the group it is in, but also how the groups perform differently in different contexts. And I think that is key as well. So some groups may do better in certain environments than others, and therefore ultimately depends in the wild what environment animals encounter, what specific predators. And that also leads to my question I asked earlier, like, if there's a lot of aerial predation but very little piscivorous predators, the grouping behavior is likely to be quite different because there's different selection pressures. With with many piscivorous predators hunting and thereby is eating fish from the edges of the group and from the back, while maybe aerial predators attack from above, and they can just attack those very sociable fish in the center. So sociability might be have a very different influence in that behavior. But that is purely hypothetical. We really need data to further investigate that.
Yeah. Thank you very much. And we
Thank you.
need to move